Discovery of dynamic self-adapting substances and their applications. This revolutionizing the drug efficacy and enabling mass-scale, self-adjusted, self-adapted dynamic medicine. I will start from modern medicine, where it goes. The slide indicates the real effectiveness of some widespread drugs and their combination in comparison with the placebo. The real effectiveness of majority of existing drugs is very low. Here we have percentage, and this is placebo. There are a lot of factors, some of them we touched today in our presentation. That's why we go to direction of precision medicine. It's one of the tendency. Would everyone buy the same clothes? Probably not. But when people get sick, they will likely receive the same medical treatment despite their many differences. The advent of precision medicine is moving us closer to more precise, predictable, and powerful healthcare that is customized for the individual patient. Our growing understanding of genetic and genomics and how they drive health, disease, and drug responses in each person enables doctors to provide better disease prevention, more accurate diagnosis, safer drug prescriptions, and more effective treatment for the many diseases and conditions that diminish our health. Tailoring health care to each person's unique genetic makeup is a very promising idea behind precision medicine. Also variously known as, they have different synonyms, kind of synonyms very close to each other, as individualized medicine or personalized medicine, despite some slight differences, or genomic medicine. Evolution of modern medicine. Traditional medicine uses one drug fits all approach based on broad population averages. The goal of personalized medicine or precision medicine is to provide the right treatment for the right patient at the right time. It considers individual difference in genes, environment, and lifestyle. However, genetic testing and personalized treatment can be expensive. A drug development involves operator we use here, this operator. Cost, time, and error. For instance, personalized medicine accumulate cost very high, time and error, and sometimes it can be unaffordable and intolerable. Traditional medicine are relatively low in cost, but have a high error rate, according to my slide I just showed. Nearly 40% of patients do not respond to static substances. Here we see precision medicine and its limitation. The key idea of personalized medicine is to develop the personalized drug for individual disease or unique person. In order to provide the personalized medicine, the patient should follow the procedure of developing personalized drugs. This procedure includes stratification, split by stratus, of risk factors, both professional and individual, personalized diagnostics of genotype, genome sequences, and genetic defects, development of personalized treatment, taking into account both unique body and unique disease. Each operation of development of personalized drugs is characterized by cost, time, and error. Error, we mean difference between needed and obtained results, just generally. Accordingly, before the personalized drugs start treating the patient disease, accumulated cost, time, and error become unaffordable, intolerable. Accumulation of the cost, time, and error in the conventional process of personalized medicine leads to unresolvable contradiction. Drugs should be personalized to provide for efficient treatment, but it should be universal to avoid intolerably high cost, time, and error. How to treat and how to resolve this contradiction. The contradiction is resolved by introducing a new generation of medical drugs. We name it dynamic drug that is universal in production and become personalized in the patient body. Universal in production. As a result, it provides for efficient treatment and avoid intolerably high cost, time, and error, because it combines both features. Drugs supposed to be self-adjusted, self-adapted, dynamic medicine. Let's go in depth how in micro-level design for dynamic medicine, philosophy and approach. First of all, let's analyze briefly and generally in images adaptability of different systems. Today we are scientists, doctors, TRIZ specialists. Tomorrow we may be patients. I schematically visualize the degree of adaptability of static classical drugs with unchangeable structure, unchangeable formula. In the human population as a blue circle with a small area proportional to adaptability. The green circle shows the effect of the human organism. It is dynamic, changeable, and it has sleeping effect due to organism aging and renewal of its cells. It overlaps the blue circle due to slippage effect. For instance, cardiologists have to change the combination of hypertensive drugs in people after a while, but their static conservative structure again falls into the blue circle because they're not changeable. From another side, microorganisms, fungi, viruses, their variability is much higher. They overlap these circles, this gray circle, of static drugs, and forces pharmaceutical companies to develop new antibiotics. Since the variability of the gray circle covers all circles, we are forced to deal with problems after they arise. For instance, to fight new viruses, it takes many years to create and approve new drugs, vaccine, whatever. But it takes many lives during this time. Since the gray circle is more flexible, the virus mutates again at the time of new drug creation, etc. It looks like a vicious circle. We don't know what is happening behind the doors of nature's lab, which is very smart. I will use one of my favorite movies, Gladiator, with Russell Crowe as Maximus, to illustrate this point. Citation: whatever comes out of this gate, you got a better chance of survival if you are ready to fight. How to resolve this contradiction? Our goal is a red circle, which shows the variability and adaptability of our proposed dynamic drugs with self-adaptation capabilities. These circles significantly overlap all possible circles, offering a more adaptable treatment approach than static drugs. Dynamic drugs adapt to a particular patient's human body, viruses, bacteria, and cancer cell even faster and more efficiently, offering a promising future in drug development and treatment. An area of this dashed circle is even would be bigger in future during development of our approach. If you look on classical drugs, we made image the principle of classical drug with a conservative structure represented like one key for one lock, one drug for one receptor. The main idea behind the creation of dynamic drugs is to use a set of lockpicks instead of one key for one lock. A group of many similar molecules that open many locks and adapt to their targets. This ensures significantly increasing efficiency and the widest possible spectrum of the drug actions. Briefly, this slide presents characteristics of drugs created with intraspecific polymorphous nature of receptors, taking into account quasi-life self-organizing system. A question arises: how should a drug that is effective for everyone look? A drug to which it is impossible for microorganism, or almost impossible, and virus to develop a tolerance. Clearly, it should be not a strictly conservative structure. First and foremost, the property of a drug like this should include the ability to change, or changeability, and the ability to adapt to external conditions. This is time so-called of dynamic drugs, we name it like this. What does it comprise? We propose that it should be a self-organizing structure based on millions of similar but varying molecules capable of forming supramolecular structures. Naturally, they should be biopolymers with irregular structure, like protein, peptides, DNA or RNA, oligonucleotides, with partially randomly mixed groups, like lysines and histidines with peptides and amino groups with oligonucleotides. The amino group substitution must be incomplete to facilitate the formation of the maximum number of derivatives that differ by level and place of substitution. Thus, in one volume of the drug, there will be millions of derivatives. You will see number even bigger than millions of the same main active ingredient that differ insignificantly from each other. At least some of the molecules will reach the necessary receptors and will be complementary to this in specific given person. If the structure of the human receptors change as a result of aging and the cell renewal process, the slippage effect, or if the virus of microorganism mutates, other fragment of the drug found in the mixture will act, and tolerance and natural selection will become impracticable. This is one of the first project, MolRex. We name it MolRex since we consider this like molecular robots. And this anti-cancer drug, it kills only cancerous cells without damaging the healthy ones. What is principle of this? Principle of selective specific hybridization of oscillation DNA, RNA. DNA temperature denaturation, separate double helix into single strands A and B. After that, we use modification, for instance, succinylation. Both single strands are acylated, reversing mononucleotide charges to opposite. Modified chain selectively hybridized only, and this is extremely important, with their precursors. For instance, chain A hybridized only with acylated chain A, as well as chain B hybridizes only with acylated chain B. This process demonstrates how charge reversal through oscillations create highly specific hybridization pattern, which could be useful in molecular biology application and targeted therapies. In this slide, we show the principle of creating anti-cancer MolRx. A yeast transport RNA, such as alanine tRNA, is taken and treated with nuclease, which lead to formation of multiple oligonucleotide RNA fragments. The resulting fragments are then modified by acylation to reverse the charges of the oligonucleotide residues carboxylation. MolRx is our novel anti-cancer drug composed of carboxylated RNA fragments. If such a mixture, this is our drug, of carboxylated oligonucleotides MolRx is mixed with tRNA, transport RNA, the complementary acylated fragment of oligonucleotides form bonds with their not acylated precursors and precipitate on tRNA. And this is extremely important. It disrupts the latter's functioning and stopping protein synthesis. No food anymore. And if you're on the next slide, you see this our drug goes through this. This is our cancer cells, and they have active macropinocytosis, selectively uptake oligomeric RNA fragments with an altered charge, MolRx, from extracellular space, leading to their demise. Cancer cell with high macropinocytosis actively, preferentially capture MolRx. Normal cells, this blue one, with little or no macropinocytosis remain unaffected. Internalized MolRx fragments interfere with protein synthesis in cancer cell. Disruption of protein synthesis results in cancer cell again. This approach leverages enhancing macropinocytosis of cancer cell to deliver MolRx, specifically disrupting vital process while minimizing the impact on normal cells. If you look at this cancer cell, and next part of our animation after they get this, our substance, illustrates the process of apoptosis of cancer cells triggered by MolRx impact on protein synthesis. No food, no life. Our experimental study, I show one of the slides, evaluate several anti-cancer drugs and bacterium-derived immunomodulatory lectin in treating multi-drug resistant bacteria. Multi-drug, it's very serious multi-drug resistant Lewis carcinoma in mice. Molrex combined with immunomodulatory lectin yield the best outcomes with 100% survival in 40 days. This one, 100%. Post-inoculation for the group of receiving both lectin in our Molrex. Molrex outperform other tested anti-cancer drugs. These results highlights Molrex's potential as promising strategy for enhancing cancer therapy, especially when combining with immunomodulatory agents. The synergetic effect suggests a powerful approach to improving cancer treatment outcomes, potentially overcoming the limitation of traditional therapies. For instance, well-known cisplatin, if you look at this, it shows about only 27% rate of survival. Okay, this one is very powerful drugs which people use today. It has a great property, and here I present some of them. It contains more than 100,000 acylated oligoRNAs that selectively target adenocarcinomas with macrophage. The acylated oligonucleotides bond with an active cancer RNA, transport, not only transport, also matrix, ribosomal, halting protein synthesis in adenocarcinoma cells. Molrex spares healthy cells which don't uptake negatively charged oligonucleotides. It demonstrates efficiency against cancer cell and adaptability to various cancer types. By exploiting cancer cell characteristic, Molrex offers potentially versatile and effective targeted anti-cancer treatment approach. For this project, we got a set of IPs, and we developed this project further. Next project is Albuvir. This dynamic drug that blocks the operation mechanism of pathogenic microorganisms. If you look at this animation, which I'll show, it depicts viral genome penetration through the cell membrane. The cell membrane surface contains nuclear pores with receptors for alpha and beta imprints, viruses, and let me make this animation. Virus poses imprinting like proteins, enabling them to activate receptors on the cell nucleus. This allows them to open nuclear pores and penetrate into nucleus. This infection mechanism is characteristic of 80% of all viruses. By inhibiting this mechanism, it may be possible to combat more than 80% of all known viral infections. It potentially leads to broad-spectrum antiviral strategies targeting nuclear entry. Albuvir reacts with peptide signals of viral imprints and block them, and the virus does not enter the nucleoli with its replicants. It can replicate because it can't enter. The most interesting property of Albuvir is the ability to adapt to many viruses that directly or indirectly use the cell nucleus. These are influenza, herpes virus, coronavirus. Albuvir demonstrates rapid onset, stopping influenza virus replication within 30 minutes. 30 minutes finished when administered orally. We had a set of IPs for this project. And another project we apply dynamic approach is dynamic vaccine, which expand immune capabilities of partially modified self-organized peptides. On this slide, After dynamic vaccine creation by partial modification of antigen, it generates thousands of variants with slightly altered surface architectonics, and it protected from enzymatic hydrolysis, its breakdown into immunogenic haptens, hapten absorbed into the bloodstream, absorbed haptens induce immune response. This approach enables oral vaccine administration while maintaining immunogenicity and overcoming traditional challenges associated with oral vaccine. For comparison, if you look at this native antigen, if it hydrolyzed by enzymes to amino acids, they don't induce immune response at all. If we apply a few modifiers, we end up with a combinatorial mixture of hundreds of thousands of proteins with minor surface structure differences. Many new immunoglobulin monoclines will be produced for such new antigen epitopes, and accordingly, the vaccine will be much more effective. During incomplete modification, modifying 2 to 10% of surface in this area, positively charged amino acid residues, for instance, for influenza neuraminidase, where we have 216 available groups for modification, leads to a mixture, imagine this number, 2 to power 216 combination of molecules that will be modified. At least one of these compounds from this variety will match the receptor of the vaccine antigen, and the vaccine will be much more effective. Remember at the beginning of our presentation, I show a big red circle. No wonder it's overlapping everything. You see this huge, imagine this huge number. One of this graph, we made experiment with Pseudomonas antigen in this graph. In this abscissa axis, we have level of oscillation as percent. And ordinate, we have antibody titer. And it shows the modification of 3%, it's about 3% of antigen, led to significant increase of the titer of induced antibodies and appearance of antibodies upon oral administration. What does it mean? So if this zero level of oscillation is regular vaccine without our process, zero level, this antibody titer is 1280. In 3% oscillation, it becomes 4 times more efficient. Can you imagine? And look at these two graphs. One graph represents what? Injection vaccine. And the second is oral. If you use injection vaccine with zero oscillation, it does not work orally. But after our technology, we can use vaccine orally. A result, in this case, when we have 3% of oscillation, is compatible with what we have in injection vaccine. This is very important. This is a way how to apply vaccine orally, since one thing, and the second, how to improve efficacy in many times of this. We have IPs for this project as well. And the next project is dynamic insulin. In this slide, show the principle of insulin dynamization. The first step involves selective trypsinolysis. These arrows show the sites of the enzyme's fragmentation. Black bars show places of insulin hydrolysis when it's treated with pepsin. Only 7 peptides are produced. Black arrows show the amino group that should be attacked by anhydride, and number of groups available for acylation equals to 17 in this case. More specifically, the process of creating dynamic insulin involves several innovative steps that modify the structure and properties of insulin. If insulin is fragmented by highly specific proteases into large fragments, which are then combinatorially modified, a mixture of thousands of acylated fragments is formed, complementary to the insulin receptors, which is easily absorbed from the intestine into the blood. To prove this concept, we made experiment on rats with alaxan-induced diabetes. A single oral administration of such a mixture of peptides stabilized the glucose level in the rat blood almost in normal level within 24 hours. See, 24 hours is almost normal. Diabetes in our and rapid and significant reduction of the blood glucose level within just 1 hour. Of administration is particularly noteworthy. We have IPs for all this project as well. And I return to, so far we made experiments. Our project, we have used our technology to create the world's first dynamic drug structures, each with unique properties for treating various diseases. However, following the mono-B-poly evolution pattern, it was logical that the following step be taken. Go from one substance to B and poly. I will demonstrate this briefly in the following projects. Schematically, we consider two cases. When we have one substance and we have different modifiers, number of modification we increase tremendously. We have set of formulas, one of this, right? Or we can have, instead of one, we can have few substances and few modifiers. And number of combination would be much more in this case. That's why next logical step was made to supramolecular antiviral systems. The dynamic self-organizing nanostructures with antiviral properties, not only antiviral, by the way. Based on experience with individual dynamic drugs, we explore combining multiple dynamic structures to create a second derivative, if we use terminology for math, nanoparticle with potentially enhanced efficacy and synergistic effect. An antiviral nanoparticle composed of multiple dynamic structures. The core, these green pentagons, dynamic cobalamin, form the nucleus. Surface, these blue triangles, represent dynamic dipyridamole, is sorbed into the core. Intermediate level, combinatorial oligopeptides, amino acid, and dynamic riboflavin embedded between core and surface molecules. This level. This complex structure was predicted to exhibit a broad spectrum of antiviral activity, potentially leveraging the unique properties of each component to enhance overall efficacy. Combining different dynamic drugs in a single nanoparticle could lead to synergistic effect, improving antiviral performance beyond what individual component might achieve alone. This approach represents a novel strategy in antiviral drug development, exploiting the versatility of dynamic structure to create more potent and adaptable therapeutic agents based on monobipolar approach. Very briefly, since we have limited time, I just show that all these ingredients we study separately, and all of them represent independent drug by itself. But combination of them give you synergistic effect. So briefly, remember this structure again. Dipyridamole, riboflavin, and cobalamin with all this stuff. For instance, dipyridamole. We use denimization process of dipyridamole derivative using a single modifier, succinic anhydride. As a result, we have 5 new derivatives which are formed. So I'm not going in depth of these molecules for simplicity, but in next slide, it was mono, right? Next is the denimization of dipyridamole with 2 modifiers, B. In this case, we have much more. Since this is symmetrical, we have 12 new derivatives are formed. The same procedure based on introduction of this presentation in the beginning, all this element we use. We took a riboflavin and 2 modifiers significantly increase the number of different derivatives of original molecules. Riboflavin modified simultaneously with succinic and maleic anhydride produce 80 derivatives reacting with each other. This diverse modification is achievable only with precise calculation of modifier quantities relative to riboflavin amount by our method. If you put any different amount, system will not work. So many years we study combinatorial, we created a theoretical combinatorial model where I can exactly count. This is our know-how. We don't represent all these details, but how we can count ingredients to have this done in this case. When multiple dynamic structures, such as cobalamin, dipyridamole, and riboflavin are separately synthesized and mixed, the resulting nanoparticles aggregate are successfully captured by both one substance, but it works for cancer and virus-infected cells. You can see here we have synergistic effect in this slide. Because nanoparticles. When you study results on HPLC chromatographs, these chromatograms of initial riboflavin and supramolecular structure and the original dipyridamole and its dynamic structure. This feature demonstrates supramolecular structure formation while preserving essential properties. You see this, many substances you have in this one. We have IPs for this project. And next example is, next our project is dynamic hemostatic device gamma. It's self-organized modified body compatible substance. And strategy for the development of binary hemostatic based on self-organized modified polysaccharides. In many complex wounds and injuries of body tissues, it is impossible to completely remove all fragments of a known hemostatic after its administration to stop bleeding. For instance, Silex or Quick Lock. In this regard, we have developed a new binary hemostat gamma, which is immunologically not rejected by the body and has the ability of better resorption and safely biodegrade in the human body. Unlike Silex and Quick Lock, it can absorb 20 times the volume of blood with the formation of a solid biodegradable gel, does not traumatize tissue during movement of the injured limb, does not cause the necrosis, and is fully immunologically tolerant to the body. The time to stop bleeding ranges from 30 seconds for small wounds to 5 minutes to severe abdominal and thoracic bleeding. Unlike Silex and Quick Lock, it can stop uncontrolled bleeding directly in the operating room during thoracic surgeries when litigator cannot be applied. The slide shows comparative data on bleeding stopping time and sorption activity of gamma and Silex. On average, from 30 seconds to 5 minutes are needed to stop bleeding, stab wounds, or gunshot wound in the limb when using our developed preparation gamma, while Silex stop bleeding in 30 minutes. The right graph shows 1 gram of gamma can absorb up to 89 grams of blood, whereas 1 gram of hitazan from Silex can absorb no more than 2 grams of blood. Absorption tremendously much. This is our IP for this project. Based what we did, I show just fragment of our research. This is evolution of medicine. From one drug for all patients to self-adjusted, cell-adapted dynamic mass medicine. This is significant departure from traditional static drug formulation. Our various projects based on dynamic drugs showcase with the versatility and potential of the innovative approach of addressing various medical needs. Rather than tailoring drugs before administration, this medication would self-personalize within the patient's body. This is very important. This allows for affordable mass production while still achieving personalized effect precision. And our drug can be applied like itself on any of the stage of modern for stratification, precision medicine, or one drug for all. Based on the dynamization idea, we solve extremely important problem which cannot be solved by static drug. Dynamic drugs has unique synergism of properties. It blocks the development of drug resistance. Remember the first circle on this slide where red area overlap all these circles. This is extremely important problem. For instance, problem for multi-drug resistant, fighting multi-drug resistant bacteria in future. It's huge problem by itself. But in addition to this, we solve many important issues. For instance, our dynamic drugs have wide action spectrum and low side effects. It's easy in mass production and affordable, with personalized property. Effective for entire human population and effective for animals. To create this kind of drugs, we started from scratch, and we created first a theory of dynamic self-adapting chemical structure and a concept for their creation, including a mathematical apparatus for calculation and modeling, which we have developed. Based on this concept, method for synthesizing dynamic chemical structure through, we name this method, partial modification of polyacceptors substances, have been developed. This innovative approach has been demonstrated to be applied to any chemical structure with 3 or more accessible groups that can be modified. For example, by carboxylation of amino groups, this structure uniquely formed with a specific ratio of components calculated according to our proposed mathematical formulas and models within the framework of the theory of dynamic chemical structures. It has been shown that proposed structures can be applied in creating dynamic self-organized and self-adapting drugs, dynamic drugs. Dynamic drugs were synthesized based on dynamic chemical structures. This drug possesses self-organizing and self-adaptation properties and can overcome drug resistance development by the human organism, microorganisms, viruses, fungi, and cancer cells. We got a set of IP and set of publications for these projects. And workforce dynamic substances, this presentation contains 30 years of research on dynamic chemical structure and their application in self-adjusted, self-adapted dynamical medical drugs, opening vast possibilities in health treatment. I began the presentation by noting that while we are scientists today, we might be patient tomorrow, driving our mission to enhance health and longevity. This led to creation of a new generation of self-adjusted dynamic medical drugs, resulting into 2 granted patents for longevity. I do not apply this, it's just fresh. Anti-aging and rejuvenation drugs. People can live longer and happily. So far, we have developed and synthesized various dynamic chemical structures, which give us extremely powerful tools for fighting disease and staying healthy. There are different kinds of represented here. Antiviral like Albuvir, Molvirex, nanoparticles, dynamic insulin, anti-cancer Molrex, binary hemostatic gamma, regeneration stimulation dipazole will help you regenerate stem cells, antimicrobial antibiotics, modified lectin immunomodulators, nephroprotectors for polymyxin, for instance, is a last resort for combating multi-drugs resistant bacteria, etc. What future do we envision? One of my favorite used in academy is the Little Prince Antoine Saint-Exupéry. Like the Little Prince box containing the perfect ship, remember, right, from Our research offers endless opportunities to those sharing our dreams. Everybody could imagine their dream of bright future in this magic box. I encompass my vision of endless innovations in adaptive medicine aimed to improve quality of life and longevity. As we age and gain experience, our wisdom becomes mankind's heritage to be passed to younger generation. For this, we must live longer, healthier, and happy. I would like to acknowledge my colleagues and friends. My colleague, Professor Artur Martinov, with whom we have done together all this research, patents, publication, with whom I have collaborated for many years, and who contribute have been invaluable. I would also like to acknowledge many of my colleagues and friends who participated in these projects, this huge project. We believe the dynamics of adapting chemical structure will help millions to be healthier and live longer. Thank you for your attention. I have more slides, but I believe this is enough for today's presentation. Thank you for attention. If you have any questions, welcome.